Now that we've spent a couple of videos talking all about the rockstar cell or the nervous system, the neuron, let's go on to the supporting players. We've talked about the neuron, the dendrites, the axon hillock, the dendritic tree, the soma, EPSPs, IPSPs, action potentials. We've kind of exhausted the neuron. So the neuron only makes up, let's say, for example, the brain. The brain is only about 50% neuron. The other 50% happens to be support cells, or what we call glial cells. These glial cells fall into a number of categories. We've got the peripheral nervous system, central nervous system. Forgive me if I'm repeating myself. This is about the fifth take on this video. Central nervous system, brain and spinal cord, peripheral nervous system, everything that goes out from there. So we need to deal with oligodendrocytes and Schwann, which are essentially doing the same thing, but just in different locations, of course, with different names. Astrocytes, ependymal, microglial, and satellite. So when we take a look at what the nervous system actually looks like, it's this. It's chaotic. It's a circus of different cells. And we've got our neuron here, dendritic tree, axon, loops around and takes off. Here's another one, axon, takes down and leaves the screen. So what are all these things? What are microglials and astrocytes and ependymals and oligodendrocytes? Let's get into this. Let me back up. Oligodendrocytes and Schwann cells, they essentially do the same thing but in different locations. And what they do is they wrap the axon in myelin. Myelin is a fatty insulation. Here is an image of a cell, an axon that's wrapped in myelin. So you can see the axon right here, and you can see all these layers of myelin that happen to be out here. The myelin might not seem like a big deal. And go, who cares? I know about the neuron. If you have or you know someone who has multiple sclerosis, the issue that they have, or you have, hopefully not you have, is with the myelin sheath. The insulation is gone. They're losing the myelin sheath. Same thing if you happened, well, if you've ever heard about Mad Hatter's disease or condition, this is an issue where when you are tanning a beaver hide or something like that, you work with mercury. Mercury tears away the myelin sheath. So it's not just the neuron that's important, it's these accessory cells, these glial cells, these support cells that play an incredibly important role. Now, myelin it wraps like this. So here's a myelin sheath, here's a myelin sheath. These are all Schwann cell derived, well, depending on which system you're in. And what we have are these little open areas of just purely axon, and we call them nodes. Specifically, we call them nodes of Ranvier, R-A-N-V-I-E-R. So the nodes of Ranvier is where we actually see the exchange of ions. Inside these myelin sheaths Not so much, because they're wrapped. It would essentially it'd be like taking an extension cord and taking the insulation off the extension cord. If you do that, the electricity is going to bleed out into the surrounding area. If you don't, it's going to move past very, very quickly, which allows essentially that action potential to jump. So it jumps from node to node to node in what's called saltitation, if I can spell it right. And that means a jumping of the action potential from one to the next, the next, to the next. Now, depending on how much myelin there is and how thick the myelin is, this is going to put you into three categories: a type, or an A, or a B, or a C. A having the most, B having middle, C having the least. Some of the C's have none. And so with a C, essentially here's what we're looking at. A C, the action potential will move either incredibly slowly or well under a meter per second. So if you compare that to my height, you're looking at maybe, if you want to go English, 5 feet per second, but that's not a big deal when you're looking at something like the brain. That's pretty fast. If I up it to a type B, now I'm looking at, and I'm going to be subjective again, bigger diameter, greater diameter, more myelin. Maybe 5 meters per second. If you think about it, that's pretty quick. And if we go up to a type A, we're talking about a lot of myelin, a big diameter, and we're talking about possibly even 100, 150 meters per second. And that's pretty fast, going from one side to the other side of the brain. So yeah, these Schwann cells, these oligodendrocytes, are incredibly important. Now there are the ependymal cells, and I don't think I'm doing this in order from what I had up a couple slides ago, but ependymal cells are simply a ciliated epithelium. So they get the cilia, they beat. Our central nervous system, our brain, our spinal cord has what's considered CNS or CNF. It's a fluid that flows around our brain and flows down through our spine. And you might wonder, well, how does it do that? These. So CFS, I got my acronym right, cerebral spinal fluid, would just sit there and stagnate. But these little cilia move it down the spinal cord, up the spinal cord, around the brain, and back down. So to keep those neurons and all the other cells healthy, it's kind of an important cell. Oh, and then the astrocytes. Astrocytes, and you can't really see it on the top of this, I wish they had included it in this image. They connect capillaries with neurons. So the capillaries are taking away CO2, taking away waste, and they're bringing in O2, they're bringing in nutrients. And so what these are doing is they're setting it up. They're transferring back and forth. Or in the case of waste and CO2, they're sending it down. Keep in mind these neurons are not mitotic. They're only focused on sending an impulse. That's it. So who takes care of them? Well, it's these glial cells. go back to this hodgepodge of different cells that are involved in the nervous system. So we've hit oligodendrocytes, Schwann cells, astrocytes, ependymals. So we've got microglials. Let me give that image. There's a microglial labeled up here. Here's one that's not labeled. Microglials are simple. They're macrophages. They're eaters. They're white blood cells. But we call them microglials because they happen to be in the nervous system. And then we have, let me go back this way, the satellite cells. Now keep in mind what I just mentioned about neurons. They can't take care of themselves. They're not dividing. What satellite cells will do is wrap themselves around neurons. When they wrap themselves around neurons, they take care of them. They make sure the temperature is right. They make sure the pH is right. So you have essentially astrocytes feeding them, taking away waste. You have satellites that are maintaining their temperature, maintaining their pH. So we kind of understand that, yeah, neurons are a big deal, but if you start pulling these out of the equation, then you're going to have a problem. So those are these satellite cells or glial cells that are a major component of the nervous system on top of or along with the neurons.